Hi, my name is Anand Sastry, and I'm a 6th-year graduate student in Bernard Paulson's lab here at UC San Diego. So when I first joined this lab, I was really interested to see how machine learning and big biological data could work together and fundamentally transform the way that we view biology. So this paper that we recently published is the first step towards that goal. So E. coli can grow in a lot of different conditions. For example, it can grow on a spinach leaf where there's not too many nutrients. It can grow in your stomach where it's highly acidic. It can grow in your gut where there's a lot of nutrients but not very much oxygen. And so it uses this transcriptional regulatory network to change the gene expression levels across the genome so that it can optimize what it's expressing for the condition that it's in. So we wanted to reverse engineer this transcriptional regulatory network, and in order to do this, we needed to get a bunch of different conditions to sample the entire space of the transcriptome. So fortunately, this lab has been doing RNA-seq experiments for a while, and I was able to gather almost 300 expression profiles that were previously published in this lab. I then took this all together and I used a machine learning algorithm called independent component analysis, and that's the basis for this study. So independent component analysis was developed to solve what's called the cocktail party problem. So imagine you walk into a room and there's all these famous people at this cocktail party, and you want to hear everything that these people are saying, but you can't listen to all of them at the same time. So you set up a bunch of microphones in this room. Each microphone picks up a different linear combination of each of the source speakers. If you go back later and you take these mixed signals and apply independent component analysis, you can actually recapture the original source signals and split them apart, and you also get the relative volumes of each of these speakers on each of the microphones. In the biological context, instead of in a room, you're in a cell, and instead of speakers, there are transcription factors and other transcriptional regulators that are sending signals across the genome. So we measured these signals using RNA-seq or expression profiling. And our hypothesis was, if we get enough expression profiles for enough diverse conditions, can we actually use ICA to get back the transcription factors and how they affect the global gene expression? And not only that, but also what is the volume or the activity of these transcription factors in each of these experimental conditions? So this method worked fairly well. We found 92 sets of genes that were independently modulated across the entire transcriptome. So we called these gene sets imodulons. This is because the set of genes that are known to be regulated by a single transcription factor is called a regulon. So when we compared our imodulons, our data-driven imodulons, to characterized regulons, we were able to directly link 2/3 of these imodulons to transcription factors. Some of them had to do with transcription factors, some of them had to do with sigma factors, which are involved in RNA polymerase recruitment. Some of them had to do with riboswitches, and a few of them measured multiple signals of different transcription factors at the same time, just because in our expression profiles, we couldn't really distinguish between the two signals. So the main outcome from this paper was actually this in the supplement. It's this 92-page PDF where each page outlines a different imodulon. So some of the information that we can get from that is what are the genes in the imodulon, what are the different activity levels of this imodulon across every single expression profile. So this kind of represents the activity of the transcription factor in that particular condition. You also get information about how well does this imodulon capture the known regulon. And in this case, for METJ, the methionine regulator, we found 6 genes that we predicted to be regulated by this METJ transcription factor that were previously not known to be regulated by it. We did ChIP-EXO, which is a way to find whether this transcription factor binds upstream of these genes, and found that in every single one of these genes that we predicted, there was transcription factor binding. And finally, you have information about the motifs found upstream of some of these genes. So this is a completely new way to represent transcriptional regulation in a cell. The results that we get from using this method on new datasets are just so clear and easy to interpret. and some of these have been published in 2 new papers from our lab. In one of these papers, we use ICA to actually show that mutations acquired in laboratory evolution can prevent DNA damage from oxidative stress. It's deep biological insights like this in complex environments that make us excited to see what ICA can teach us about infections and pathogenicity for bacteria. So we're currently building up our software so that we can analyze even more expression profiles from publicly available data, and also expand this analysis to all sorts of different organisms like Staph aureus and other infectious diseases. We're really excited to see the type of insight that ICA is able to bring to these organisms.